Hello everyone, very warm welcome everyone to ITLS Academy. Our lecture is continuation part of additional applications of microorganisms in the pharmaceutical science. In our previous lecture of additional applications of microorganisms, we have seen various kinds of microorganisms and also many products produced by these microorganisms like vitamins, organic acids, and enzymes, etc. But that lesson was not sufficient to study all the informations for the additional applications. So left out things we will see in this particular lecture. So let's start our part 2. Today we will see applications of microorganisms in the partial synthesis of pharmaceuticals. The very first point is whole microbial cells as well as microbially derived enzymes have played a significant role in the production of novel antibiotics. The potential of microorganisms as a chemical catalyst or enzymes, as we know that enzymes are catalyst, enzymes work as catalyst. So however, first fully realized in the synthesis of industrially important steroids. We can say that when the industrially synthesized steroid was developed, then the full potential of these microorganisms was seen. So now let's see production of antibiotics. The point is, in the antibiotic industry, the hydrolysis of benzyl penicillin to give 6-aminopenicillinic acid by the enzyme penicillin acyclase is an important stage in the synthesis of many clinically useful penicillins. Next point. A second example is provided by the production by fermentation of cephalosporin C, which is used solely for the subsequent preparation of semi-synthetic cephalosporins. Furthermore, antibiotics produced by fermentation of various molds or especially Streptomyces species, and this can be employed by medicinal chemists as starting blocks in the production of what might be more effective antimicrobial compound. Next comes steroid biotransformations. First point since steroid hormones can only be obtained in small quantities directly from mammals. Attempts were made to synthesize them from plant sterols, which can be obtained cheaply and economically in large quantities. Although it is easy to hydroxylate a steroidal compound, it is extremely difficult to obtain site-specific hydroxylation so that many of the routes used for synthesizing these desired steroid are lengthy, complex, and consequently expensive. Next point, more recent advances involving the employment of microorganisms in biotransformation reactions utilize immobilized cells. Whole microbial cells contain complex multi-step enzyme systems, and there is therefore no longer a need to extract enzymes or enzyme systems which may be inactivated during purification procedures. You can see here a conversion of, in this diagram we are talking about this particular figure, conversion of progesterone to 11 alpha hydroxyprogesterone by Rhizopus nigricans. You can see this is a chemical structure of progesterone. This is a 4-ringed structure. 3 benzene rings and 1 pentameric ring is present in progesterone. When Rhizopus nigricans act over this progesterone, it converts it to 11 alpha hydroxyprogesterone. The work of Rhizopus nigricans is to hydroxylate this progesterone on 11th position. You can see hydroxyl group is attached is 11th carbon. This is 11th carbon of progesterone, and this Rhizopus nigricans adds a hydroxyl group on this particular 11th position and converts it to 11 alpha hydroxyprogesterone. Next is chiral inversion. First we will see what is chiral. We see if there is a carbon and it is attached to 4 groups, if these 4 groups are common, like if this is hydrogen, hydrogen all around, all 4 groups are hydrogen, then it will not be chiral. While if this carbon has 4 different groups, like we can draw, this is hydrogen, this is ammonia group, this is any particular R group, and this is carboxylic group, that is COOH. You can see this carbon is attached to 4 different groups. All groups are different groups, not same. So this carbon will be known as chiral carbon. So chirality is something having all valencies filled by different groups. So now we will discuss about chiral inversion. Several clinically used drugs, for example, salbutamol, propanol, and 2-aryl propionic acids, in short NSAIDs, are employed in the racemic form. Next point, in the last series, for example, buprofen activity resides almost exclusively in the S isomer, as we know that compounds exist in two forms, that is R and S isomers. So in the last series the activity most of the time favors S isomer. Chiral inversion in the unidirectional manner, that is from R to S, when R form converts into S form, this is known as chiral inversion, and this occurs in vivo over a 3-hour period. This is the time period that is utilized for the chiral inversion. Enantiomerically pure S form could be administered clinically to give a reduced dose and possible less toxicity. You can see here this is chiral carbon. This is attached to 4 different groups, as this is hydrogen, this is argon, this is methyl group, and this is carboxylic group. So this carbon will be known as a chiral carbon. The form of this carbon is R form. These all groups are in the R form, while in the case of S form you can see these are converted into S form. For the S form, here the order of the functional groups are changed. You can see these are arranged in a proper form, that is having less atomic weight, more, and more atomic weight. These are something in a corrected order, and this is S form. Now we are going to see use of microorganisms and their products in assay. So the point is, microorganisms have found widespread uses in the performance of bioassay. For the very first point, for determining the concentrations of certain compounds, for example amino acids, vitamins, and some antibiotics. And this determination of concentration occurs in complex chemical mixtures or in body fluids. Next point, for diagnosing certain diseases. Next is for testing chemicals for potential mutagenicity or carcinogenicity. Fourth point is for monitoring purpose involving the use of immobilized enzymes. And the fifth and last use of microorganisms is sterility testing of antibiotics. Here you can say bioassay of antibiotics. So this is antibiotic bioassay. Antibiotics may be assayed by a variety of methods. You can see two methods are given by the graph. First is graphical method. Although both are graphical methods, first is representation of a 2 by 2 assay response. Here x is the horizontal distance between two lines. You can say these are horizontal lines, and here this x is the distance between these two horizontal lines. These are lines and this is a distance. The anti-log of x gives the relative potency of the standard and test. For the next bioassay, here you can see relationship between concentration of aminoglycosides, antibiotic, and the transfer of radioactivity from adenosine triphosphate to phosphocellulose. Here you can say in the first graph, zone diameter is given on the y-axis while the antilog of x is given on the x-axis. In the second graph, gentamicin concentration is given on the x-axis while counts per minute is given on the y-axis. These graphs are of antibiotic bioassay. So now we will see microbiological assays. First point, in microbiological assays, the response of a growing population of microorganisms to the antimicrobial agent is measured. We can say that microbial agent is measured in microbiological assays. In the commonest form of microbiological assay used today, samples to be assayed are applied in some form of reservoir. like uh, percolin cup, paper disc or well, to a thin layer of agar seeded with indicator organism. Next point, during incubation, the antibiotic diffuses from the reservoir and that part of the microbial population away from the influence of the antibiotic increases by cell division. The position of the zone is thus determined by the initial population density, growth rate of the organism and the rate of diffusion of the antibiotics. Next point is in microbiological assay is conventional plate assays require several hours incubation and consequently the possibility of using rapid microbiological assay methods has been studied. Two such methods of microbiological assays are Urease assay and luciferase assay. So now we will see these assays in detail. So first one is Urease assay. When Proteus mirabilis grow in a urea containing medium, it hydrolyzes the urea to ammonia and consequently raises the pH of the medium. This production of urease is inhibited by aminoglycoside antibiotics, and these aminoglycoside antibiotics are inhibitors of protein synthesis. In practice, it is difficult to obtain reliable results by this method. So, we switch to our next assay, that is luciferase assay. In this technique, that is in the luciferase assay, firefly luciferase is used to measure small amounts of adenosine triphosphate, that is ATP or energy packet, in a bacterial culture. ATP levels being reduced by the inhibitory action of aminoglycoside antibiotics. This method of microbiological assay may find more applications in the future as more active and reliable luciferase preparations become available. So now we will see radioenzymatic or transferase assay. First point is these depend on the fact that bacterial resistance to aminoglycosides such as gentamicin, tobramycin, amikacin, natilmycin, streptomycin, spectinomycin, etc. and chloramphenicol is frequently associated with the presence of specific enzymes often coded for by transmissible plasmids which either acetylate, adenylate or phosphorylate the antibiotics, thereby rendering them inactive. If we talk about the acetyltransferases attack, so acetyltransferases attacks susceptible amino groups and this require acetyl coenzyme A, while AAD or APH enzymes attack susceptible hydroxyl groups and require ATP or another nucleotide triphosphate. Next point is several AAC and AAD enzymes have been used for assays. The enzyme and the appropriate radio-labeled cofactor are used to radio-label the drug being assayed. These types of assays are rapid, taking approximately 2 hours, show good precision and are much more specific than microbiological assays. So we can say the radioenzymatic or transferase assays are preferable over microbiological assays. Although we have studied all these assays. So thank you guys for watching this session. Follow ITLS Academy on Instagram, Facebook, Twitter, YouTube, WhatsApp and also on LinkedIn. For latest videos and updates by ITLS Academy, subscribe ITLS Academy on YouTube and also do not forget to press the bell icon so that you can get an notification whenever there is an upload of any video. For easy access of ITLS Academy, get it on Google Play Store. Thank you for watching. If you like this video, then like it, share with your friends and also subscribe ITLS Academy. For any help or query, you can call us our helpline number 7080-833-450. You can also reach us on our website www.itlsacademy.com. So thank you very much guys for watching this session. Stay connected with ITLS Academy and stay safe.